Hello, Bio30. This is topic 6, mutations. So there's going to be two types of mutations, chromosomal and point mutations. The first one, chromosomal, deals with the number of chromosomes. And if you remember, the normal number of chromosomes should be 46. So if we have 45, that's going to be a mutation. If we have 47, that's also going to be a mutation. And you can see here on this karyotype, you should have pairs of chromosome because you get one from mom and one from dad. But you can see at number 21, there's 3. And so this karyotype has 47 chromosomes, and that would be a mutation. The next one here, point mutation. Well, this is actually changes on the DNA itself. So going back when you guys did DNA, you remember there was 4 base pairs, and they complemented each other. A went with T and C went with G. And then during reproduction, they also met up in a complementary fashion. What would happen then if the original base was switched out with a different base, substituted? Or what would happen maybe if a base was deleted or a base was added? That would result in a different amino acid and therefore a different protein, and that would be a mutation on the DNA itself. What would cause a mutation? Well, these are just some examples. These are mutagenic agents, maybe radiation such as X-rays or UV radiation or microwave radiation. Maybe certain chemicals such as cigarette smoke can also do this. Now, looking at chromosomal mutations, you can see in this karyotype, remember that the normal number should be 46, so there should be 23 pairs. Looking at the karyotype on the right side, you can see that the sex chromosomes, that there's not 2, there's actually 3. So again, this would be Klinefelter's syndrome, and that would have 47 chromosomes, and so that would be a chromosomal mutation. So here's another karyotype here. Remember that karyotypes are arranged by size, so the size of the chromosome, the banding pattern, so the location of the genes should match up, and then where is the centromere, so the position of the centromere that will hold those chromosomes together when they are replicated. How do we get the karyotype? We may talk about this a little bit more in reproduction, but they can do a karyotype on a fetus, and you can see here that there's 2 different ways. You can do what's called a CVS, or a chorionic villus sampling, where they will take, you can see here, a little vacuum tube, and they will scrape the outside of the membrane, known as the villus of the fetus, and collect tissues that way. And in the tissue, they will collect the cells and the DNA and then do a karyotype. This would be done maybe, at about 8 weeks of pregnancy. And then the bottom one here you can see is called an amniocentesis, where they would actually just take a needle and collect the fluid that is in the amniotic sac surrounding the fetus, and take the cells and the DNA out of the fluid. Once they have the DNA out of those cells, they would centrifuge those cells to separate them into layers. The white blood cells would be transferred and treated, and then we would say, during metaphase, you can see here, we would fix that sample and stain it and then spread it on a microscope slide. We then would see the diploid cells, like on those pictures, and we can see the chromosomes, and then we could count them up and make sure that there is 46. Now, what would happen if there wasn't 46? Well, non-disjunction is the process that may happen, and that would be abnormal meiosis. And so during anaphase 1 or anaphase 2, when the chromosomes are separating, you may get unequal separation. And so instead of example, 23 going to one side and 23 going to the other side, maybe you would get 24 going to one side and 22 going to the other, and that would be unequal separation and result in a cell, or in this case a gamete, that has either more chromosomes or less chromosomes. So here's a great picture that's showing that. And you can see here on the left hand side what we just said. Normally you should have 46 chromosomes like in the cell at the top. However, when that cell starts to do meiosis and undergoes separation, non-disjunction occurs, and then when they separate, you get 24 on one side and 22 on the other. Therefore, when meiosis is followed through and completed, you can see that you're going to have two gametes that have +1, and then two gametes that have -1 chromosomes. Maybe it happens during anaphase 2, and so you can see on the right-hand side, 46 chromosomes at the top, which is completely normal. And then it goes through anaphase 1, which is okay, but then during anaphase 2, when it separates again, you may get unequal division, which this would be less severity, because now you would get only two gametes that are not equal, so you get +1 or -1, but then the other two would be fine, so they're both at 23. So just to recap, non-disjunction occurs during anaphase 1 and during anaphase 2. If non-disjunction occurs during anaphase 1, all of the gametes are affected, and you would have two gametes that have one extra and two gametes that have one less. If non-disjunction happens during anaphase 2, only 50% of the gametes are affected. The other two are unaffected. One would have more, one would have less. This is how you might get trisomy or monosomy. So there was a karyotype when I said there was 3 chromosomes at location 21. That's known as trisomy 21 or Down syndrome. Monosomy, maybe we're missing a chromosome, such as example would be Turner's syndrome, where we're missing a chromosome at number 23. So in your book you have some questions that you can do. And now we're going to talk about point mutations. So remember that these were mutations that were on the DNA itself. So maybe, the cell has 46 chromosomes. However, on one of those chromosomes, there's a base that was changed. So looking at this slide, we can see on the top left, A, we have normal DNA. So the sequence is normal. However, maybe during reproduction or something happening, you can see the second base, which was a G, is changed to a C. It's swapped out. Well, if we swap a base 1 for 1, that's called substitution. And you can see that will actually result in a different amino acid. And if we have a different amino acid, we will have a different protein, which codes for something different. Now, maybe an example would be a protein for blue eyes. Well, if this was to happen, maybe now your mutation is one of the eyes is a different color. Down at the bottom, you can see there's a couple more types. They're called frame shift mutations. This is when we can add a base, or maybe a base gets deleted. So if you remember going back to the normal DNA, so it should be CGA. However, if we add a base, now it's CCGA. So there's an extra base there, which then will code for different amino acids, therefore resulting in a different protein. Everything in this case, though, has shifted to the right. As soon as we add that base, all of those other bases shift to the right. And if we remember when we make proteins, we read those bases 3 at a time. So that means every amino acid after that addition is going to be different. The same may happen if we delete a base. Everything shifts to the left. That's why these are called frame shift mutations. And if everything shifts to the left, then that means all of those amino acids from that base getting deleted will also change. If we're looking at mutations in this case, the most severe mutations will be frame shift mutations because of all of those amino acids getting changed, versus the substitution one where only maybe one of those amino acids gets changed. Okay, so you can see here, substitution is a mutation that exchanges only one base for another base. So it's a 1-for-1. Okay. Versus the frame shift mutation, where if we add or delete a base, then all of those other bases are changed. So here's an example of a frame shift. If we look at this sentence, the fat cat sat, if we were to take the T away and everything shifts to the left, it changes all of those words. So if that had been DNA bases, you can see how all of those amino acids would have changed. So here's the example. This one's in your notes. You can see in the yellow here, it's the original sequence, the original mRNA sequence. In green is the original DNA sequence. And then you can see underneath that the original amino acid sequence. Number 2, 3, and 4 are going through the mutations. Number 2 is what would happen if we substituted a base. So it says change the first G to a C. So looking at the green, you can see we have TACGTT, which now down here we have TAC, and then that first G gets changed to a C. Well, if we do that, that means then the mRNA gets changed to GAA instead of CAA, and instead of the amino acid glutamine, if we look on our chart, it now changes to glutamate. However, look at all of the other amino acids, you can see that they stay the same. Versus number 3 here, let's go to the addition of a T before the first A. Well, the first A happens in the second base in the green. You can see there TAC. That's my original sequence. So if I change that A now to a T, now I have TTA. Well, that means then all of the other bases are going to get shifted to the right. If they all get shifted to the right, that means my mRNA sequence is going to be completely different. And you can see it results in completely different amino acids and therefore a completely different protein. Number 4 says deletion of the first G. So you can come back up to the green, TACGTT. Well, now I'm going to delete that G. That means everything shifts down to the left. And I'm left with looking at number 4, TACTTT. And then CAG, AGA, and then CT. So all of those amino acids are then again changed as everything shifts to the left. So your idea now is to go back into your notes and identify what you think the mutation is, if it's a substitution, deletion, or addition. And I'm going to finish off here talking about the variables. These variables do come up on the exams and the diploma, so I'm just going to quickly review. If we're talking about variables during an experiment, there's these common 3. Manipulated variable. Manipulated meaning what did I change? So what is the thing that we're changing or the thing that we're testing on? The responding variable would be, well, what are my results? This is usually what you would put on a graph. And then the controlled variable would be, well, what stayed the same? So if I use the example, I don't know, maybe we're looking at measuring the heart rate, and I'm going to give half the class water, and I'm going to give the other class an energy drink. My manipulated variable would be the energy drink because I changed it from the water. The responding variable is, well, I'm trying to measure the blood pressure from taking the energy drink. That would be my results. The controlled variable would be maybe the temperature stayed the same in the room. Maybe the lights stayed the same in the room. Maybe the furniture stayed the same in the room. Okay, all of those things in the room that we didn't change and stayed the same would be a controlled variable. So you can read through, these are some examples in your notes of what the variables are. And that's the end for topic 6, mutations.